Alright, my girls. So this is a little bit awkward because I mostly have no idea what I'm doing. And probably all of your teachers have started their screencast with the exact same comment. So these videos or screencasts, I can only make them in little 15-minute increments. They don't let me make longer than 15 minutes at a time without paying for it. So I'm going to try to keep everything that I do in little 15-minute increments. Hopefully we won't have to do too many where you watch like one or two in a row. Hopefully it'll just be one and then whatever assignment goes with it, you can do that. But I'm just asking for a little bit of grace because again, I have no idea what I'm doing and I've pretty much already been rambling for 50 seconds, which is really weird because the little recorder at the bottom of the screen just sits here and tells me that I've been talking for 57 seconds now. So I'm going to try not to watch that and psych myself out. So here we go. So we left off here talking about DNA replication. Now, first hour, you're probably quite confused because we started with slide 1 of this PowerPoint. And when I realized how confused you guys were for second, third, fifth, and sixth hour, I started with this slide and they seem to understand it a lot better. Second, third, fifth, and sixth, please feel free to re-listen to this. It doesn't hurt to re-listen to it. But I think most of you have kind of already got a good grasp of what's going on in this particular slide. So the rest of you, first hour, if you would like to continue watching or should continue watching, I'm kind of going to go over what all is going on in this overall picture here. So these black lines that you're seeing are representing the old DNA or the parent DNA strands. And what you'll notice is that one of them has the 3' end up here in the top left, and the other one has the 5' end down here on the bottom left. So we know that our DNA strands are anti-parallel, right? Don't forget that word. So they're anti-parallel. So we always have to maintain that anti-parallel nature even when we are replicating the DNA. So the new strands that we're building, which are being shown in blue, so the blue strands are the new DNA that's being made. Okay, so those new strands are also going to have to maintain that 5' to 3' nature that we've talked about. So if you look again at the top left, over here where my little pointer is, so if you look up here, you can see that if this guy is the 3' on the old strand, then the new strand is going to have to start with the 5' right there, okay? And we'll talk a little bit more about why this guy's the leading strand in just a little bit. And then down here, you can see that this old strand, this is the 5' end, and the new the new DNA strand, right? This one starts with the 3', right? Because if this one is 5, then this one must be 3. So we call this guy the lagging strand because of that 3' end, but we'll talk about that in just a second. So I'm going to erase everything that's on the screen because it's looking a little busy and I don't want to confuse anyone, and when I'm in presentation mode it doesn't let me change the color. Maybe it does and one of you guys know how to do that. If you know how to change the color in presentation mode, please feel free to Skype me and let me know how to do that because I really don't like red. I'd rather write in pink, but here we are. So let's talk about what all of these enzymes are doing. So all these colorful guys that are going on really over here in the right half of our picture. So helicase over here, his job, like you guys have all told me a million times, is to unwind the DNA. So he's unwinding DNA, or he's breaking the hydrogen bonds. Also, sorry if you can hear my cats screaming at each other in the background. Breaking hydrogen bonds. Okay, so helicase's job is to move down the DNA strand, okay, down that parent strand. And over time, he's going to open up all of those hydrogen bonds. Okay, so that's helicase's job. I think everyone was pretty good with that on Friday. So the next guy I want to talk about are these green ones. Okay, these green ones are called the single-stranded binding proteins, or the SSBPs for short. That's not really much shorter, but we'll go with it. Okay, so the SSBPs are the single-stranded binding proteins. Now, their job is to basically hold the parent DNA open long enough that the DNA polymerase that adds the new nucleotides actually has enough time to come in and add nucleotides before the parent strand or before the old strands really try to come back together, because those hydrogen bonds, they don't want to be broken. Those A's and T's want to come back together. Those C's and G's want to come back together. Chemistry is pulling them together. It's attracting them. So the single-stranded binding proteins are there to make sure that the old strands stay separated long enough that DNA polymerase can come in and add in the new nucleotides. Okay, remember that nucleotides are the monomers of nucleic acids. Don't forget that. Okay, monomers. I feel like Sal from Khan Academy. Monomers of nucleic acids. Okay, and so the single-stranded binding proteins are holding open the parent strands. Okay, so that's their job. And it feels really weird to not be able to stop and ask you guys if you have any questions. So hopefully I'm doing a good job. Still kind of have no idea what I'm doing, so that's fine. I hope you guys are enjoying your time off. I don't even know if I'd consider it time off, but, you know, I hope you're enjoying it. I have been eating all of my quarantine snacks already, so it's not very good for me. Alright, so the next guy that I want to talk about is this guy over here, topoisomerase. Topoisomerase. So his job is to make sure that all of the DNA over here that isn't opened up yet by the helicase, his job is to make sure that the DNA there doesn't wind up so tight that you can't, that the DNA breaks. So if you, and I did this in 2nd through 6th hour, but I didn't think to do it in 1st hour. I think we might have run out of time. I think I might have handed the beads out, but we didn't have enough time. But if you pick up a Mardi Gras bead, if you have one lying around the house, pick one up and twist it so that it makes like a double helix of DNA, and just like twist it like you would if you were just playing with it in your hand. So if you twist it up, and then on one side of it, use one of your hands to kind of start opening up the bead while the rest of it stays twisted, you'll notice that the twisty side that you're not untwisting yet, it gets really, really tight. And when it gets really, really tight like that, it's easy for your Mardi Gras bead to break. It's the same idea with your DNA. If it gets too tight over here on this side, if that gets too tight, then the DNA is going to essentially break. Those phosphodiester bonds are going to start breaking. And if your DNA starts breaking, no bueno, that's when you can get all types of genetic mutations or proteins stop being made that are necessary for really vital functions like cellular respiration that we've talked about. So you don't want your DNA to break. So topoisomerase essentially acts like a masseuse for the DNA to make sure that it doesn't break over there. So now what do we have going on? So I've talked about helicase, so we can check that one off. We've talked about the single-stranded binding proteins, and we've talked about topoisomerase. The next guy I want to talk about is, let's go with primase. So let's go with this guy right here. So primase has a pretty cool job. So basically he's super duper important, and if you don't have primase, you're not going to be able to replicate your DNA, and if you can't replicate your DNA, then that cell cannot divide, and if that cell cannot divide, then if that would be, for example, like the zygote where the sperm and egg come together, if that sperm and egg that come together are missing the gene that codes for the enzyme primase, then that zygote is probably, or not probably, won't end up developing into an actual baby because that cell won't be able to divide. And cell division is vital to making you go from one cell to the 3 trillion cells that make you up now. So primase's job is to come in and lay down an RNA primer. So that's what this red line is showing you right here. It's an RNA primer. So RNA primers are going to be basically the cousin to DNA. So RNA is ribonucleic acid. I know we haven't talked about him yet, but he's ribonucleic acid. He's still made of nucleotides, the same way DNA is made of nucleotides, but his sugar is ribose instead of deoxyribose. But primase comes down and will lay down a RNA primer, so just a couple of nucleotides made up of ribonucleic acid instead of deoxyribonucleic acid. Don't ask me why. I don't know why it's this way, but this is the way that it is. So he'll put down the nucleic acid, the RNA primer, and from there, DNA polymerase, so there's kind of a lot of them that are functioning at the same time. This guy is proofreading. We won't really talk about him. That's more of an AP level thing that you would need to know. But his job is to proofread. DNA polymerase 3, and you don't need to remember the 3, you can just remember DNA polymerase. But he's the one that is going to add new nucleotides onto the 3' end of DNA. Remember, he can only add to the 3' end. That's it. All he can do. Okay, so not only can he only add to the 3' end, but he can't start from scratch. He always needs an RNA primer. Again, don't ask me why. I don't know. Can't start from scratch. He needs a primer. Always. He cannot ever start from scratch. And so primase over here puts down the RNA primer that DNA polymerase needs to work from. Okay, and I'm going to stop this little screen recording right here because I see that I only have like 2 minutes left, and obviously there's no way I can tell you the rest of this in 2 minutes. So I said in the beginning that I was going to try to keep these under 15 minutes, and I promise I'm really going to try. But this one is just, this is an important process and you really need to understand what's going on. So sit tight and I will upload the next video right behind this one, so you'll have it to watch.